Is this a sponge, Steve?
It is a tunicate, I believe.
Tunicate. Okay.
I think they're from the family Chordata, right?
Yeah, that's right. Yep. I've seen something like this recently, and
Go wide for a second.
everyone was a bit confused.
Okay.
As to what it could be, whether it was a hydroid or something. Let me double check.
You can zoom back in now.
Sure. Big right there.
Yeah, perfect. Giant dust cloud catching up with me. Oh, there, Steve?
Yeah, that's perfect, yeah. Since these is a colonial animal, leaving some will allow it to regrow.
So what do we hope to find in these in this sample?
Hoping to identify it first and foremost, because I think that tunicates are pretty poorly collected from this area, especially the most gelatinous ones. Oftentimes they won't come up intact if it's been like trawled or dredged over the past.
Suction at 75.
Right. So are we hoping to collect it and then preserve it and then identify?
That positive
yes,
75.
Yeah, and we don't have any tunicate
75.
specialists on board, so we'll have to reach out to some folks who are most interested in these, and they can request those materials from the museum. We did collect a tunicate, predatory tunicate on one of the rocks last night. How did that come out? Was it kind of just like gelatinous ooze?
Yeah, basically a small blob.
Yeah.
As soon as you see it in the jar.
The interesting thing is that this
suction and then rotate.
This colonial tunicate's not present in our ID guides, which is
024 is in jar 1.
Success.
Jar 4.
Jar 4. Thank you.
Not being in the guides suggests that this has not been collected or identified from this area, so it's a very useful, valuable collection.
So you said a carnivorous tunicate?
Nice grab, Dan.
Carnivorous, yeah, something we collected yesterday.
Huh. So how does that exactly work? How does something like this be carnivorous? Does it have teeth, a digestive tract? How does it work?
So if you're familiar with the Venus flytrap, right? It's a plant, it has neither of those things that you just mentioned. But it can trap prey using its musculature.
Yes. Yeah, you can.
And digest them within its within its body, within its tissue. So it kind of looks like a sock puppet, if you will, and then they'll trap maybe small crustaceans or some animals that whatever it preferentially preys upon, and digest them slowly.
Wow. And they just dissolve it with, what you said, lipids?
Yeah.
Wow.
I'm not sure of the actual mechanism they use to, some sort of digestive juices.
And then it just dissolves their prey?
Yeah.
Wow.
And then they slurp it up and consume the chemical energy that they contained in the animal.
Note to self, do not mess with carnivorous tunicate. So how big can these things grow, usually, these tunicates?
The carnivorous ones?
Just tunicates in general. On average, how big can they grow?
So some of them can get pretty large. There's one called a horsehead tunicate that can get, it's on a very thin stalk, but it can grow a couple meters tall sometimes.
Oh, wow.
Yeah.
And most of the time, aside from the carnivorous ones, these feed off of this marine snow, this detritus floating around?
Yeah, most of them are filter feeders.
Atlanta heading 350.
Roger.
was a good find. I've seen something like that before, but I don't think it's been well identified in this area, so definitely a worthwhile collection.
Coming around to 030.
Copy, thanks.
Plenty of room for rocks if you ever want to grab another one. We can always double up. We don't have to take 10. We can do.
You
one
can never
or two.
have too many rocks.
Okay, let's take one then at this outcrop. Whatever looks most promising.
Right. If we, well, if we see one, I suppose. It's a little critter down there.
Something's jumping, yeah.
Yeah, just to the right of the lasers.
I imagine the slope will start to pick up again in a little bit too. So it'll probably have more material if nothing looks good here.
Try to zoom in there.
Yeah,
See if we can catch an image of it.
Oh, yeah.
Wow.
Nice.
Is that a polychaete?
It is. Yep.
Is that the same polychaete we saw in the cucumbers?
Probably different. So this one is my families are pretty bad for polychaetes. I wonder if it's well, actually we can get a better look at it now. Oh, never mind.
No, I
bye.
touched and it scared him. He
It was
ran
between
away.
a couple of families. It looked like a bristle worm.
Steve, here's a question for you. What's the difference between a kebab sponge and a tunicate?
I don't know what a kebab sponge is. I have never heard of something like that, but I
Tunicate
have
did
a
look like a
picture in
kebab.
my mind of what I think it could be. But they're very distantly related in invertebrate animals. Sponges are very rudimentary critters, whereas tunicates go through a very complex life cycle. They're similar in that they're both either filter or suspension feeders, but otherwise they have very different processes that they go through for development. Sometimes feeding. They both probably have similar morphologies. You know, it could be some sort of convergent morphology, meaning, you know, they have, you know, they grow to similar, they've evolved similar morphologies because it suits their environment. Yeah, but yeah, I'm not sure of the details.
What did we have yesterday for a 6/8 wrap? It was a negative one? That note you put in the book from yesterday?
That's correct. 1 negative turn on the 6/8.
So you can zero the 6/8
Although
wrap
I would
there.
appreciate if shoreside folks could refrain from using food-related
you can make
jargon
a note
in
the
there.
book that way.
Animals identifications.
We're
There's
another
one.
Remove
2 hours
the
before we can get
positive turn
breakfast.
from
Yeah.
yesterday.
I know everyone here is.
Was that another bamboo claw?
Now all the chat's going to be flooded with food, and now I'm going to have to be forced to look at it. These rocks are all rounded.
Yeah, nothing looks great here.
Yeah, for tunicates, their development is really interesting since they're in like one of the latter evolved phylums of Chordata, but they don't really have like a spinal cord per se.
Yeah.
So then how do they keep that sort of that structure that we see, that long line with all those tubes on it if they don't have like a structure, a skeletal structure?
That's a very good question. Kind of the characteristics of a organism that's in phylum Chordata is a hollow dorsal nerve cord, I think. Pharyngeal gill slits, and I know I'm forgetting a couple others, but I think that may be what we're seeing in the tunicate that we saw. But that is a very good question, how it kept that branched formation.
It's probably, you know, they don't calcify as far as I know, so it's probably proteinaceous, you know, some sort of proteinaceous structure that helps the colony grow. But most tunicates, you know, they retain those characteristics like the notochord in the larval stages, and that's eventually lost or reabsorbed. You know, whereas in a vertebrate it would develop into effectively a spinal cord or nerve cord or something like that.
I see.
So they would have it during a larval stage and then, you know, subsequently lose it when they settled.
So would they be technically considered invert during an adult phase?
Yeah, I mean invertebrates, you know, the whole time. I think, you know, vertebrates really refers to the calcification of the dorsal nerve cord.
I see.
You know, so vertebrae, you know, the bony structures.
Gotcha.
Or something resembling bony structures. Could be cartilaginous or something like that too.
Oh yeah.
Atlanta heading 085.
Roger, 085.
Come up a little bit here.
Atlanta's presenting an interesting sonar target. I think we'll go have a look at. Sure.
Another Chyrsogorgia.
Got a little sea snow with us. do a quick zoom on that one as we go by, if you want.
Sure thing.
So would that be a golden coral?
There we go. That's right.
Beautiful.
And are those squat lobsters inside of it?
Yeah, at least a pair of them, I think. Sometimes you'll have a mating pairs, so one male and one female live in the whole colony. Sometimes it'll be more than, two, multiple generations maybe.
Do they ever wander outside of
Okay.
that home?
Thanks.
No, they won't. They will hold on there for life.
Wow.
We've sampled some animals, some corals, and during the process of trying to stow them, you get blown around quite a bit, and those things will hold on there for just about anything,
Dang.
almost like through hurricane force.
Impressive.
Water velocities. They are very good at holding on. Once they find a spot, they will stay there. They will guard it.
And you say generations can live in there, so like if the home is, well, filled to capacity, would they have to migrate to another home?
Yeah, that's a good question. I would imagine, that probably happens during the larval stage, when they're in the plankton. They probably use those,
there,
if there aren't the right settlement queues to settle in their home colony, they'll probably migrate. It's very there we go, dangerous for these kind of soft-bodied crabs that just walk across the seafloor. You rarely see them free living like that. So it's all probably done in the plankton and the very young larval stages.
Okay.
That is something that would be interesting to look at, would be population genetics of, crabs that live associated with corals across the seamount landscape, or even between seamounts. What's their genetic similarity or dissimilarity like? How closely related are they? Do some parentage analysis.
Ooh, that'd be exciting.
Yeah.
Would you think that'll also give us a clue about like the current flow at the at these depths?
It could, yep. Could do some inference.
And this piece of coral coming up, is this more bamboo coral? Is this...
Swinging around to 110.
That's what I would say, yep, looks like it.
I'm learning.
You're all going to be pros by the time we're done here.
Aye.
Yeah, it looks like a bamboo coral, unbranched. We haven't taken any water samples yet, but I'm hoping to see like more than one coral in a frame before I pop that first Niskin.
Is that what we look for specifically in water samples?
Well, you want a relatively high density, or numerically high density in an area to maximize your potential of getting good eDNA.
Here's another question for the chat. How would someone go about getting current data from these dives? Or do you have to wait till afterwards?
current as in like flow speed of water current, or like data from the current expedition?
I think it's data from the current expedition.
Okay. Couple ways of doing that. There is a majority, there's a ton of data that is freely available shortly after the cruise from multi-beam bathymetry that Nautilus takes during mapping expeditions and during site selection and scanning for ROV cruises, up available on a site called R2R, or Rolling Deck to Repository. That's hosted also in other repositories that are managed by the government, NCEI, I believe, National Centers for Environmental Information. In addition, things like the samples that we collect are available to scientific community through our partnering geological laboratory. Can we tilt up a little bit when you are done in front here?
Want me to come up? Where did it go?
So there's something over in this area. Can we move over there more?
Sorry, they meant water current. How to get the data from the water current.
Right there.
Yeah.
Oh, wow.
Oh.
Take a look at that. I think it's a sponge stalk.
Oh yeah, I see it now. Oh,
There's
there's also
A brittle
yellow
stalk.
crinoid.
Oh, that's a card. Oh,
Um,
wow.
Current data. So we don't really track water flow using any seafloor vehicles or any sensors on the seafloor. And we don't really have the equipment on Nautilus to monitor current conditions from the surface. You would need something called ADCP, Acoustic Doppler Current Profiler. Although, even though we don't have those, we can still, most of what we do is make inferences based on what we're observing in situ. So which way currents are flowing on the seafloor. We also use some databases that use models to generate, current conditions in the seafloor.
Sorry,
one
Steve,
of those. Yep.
I'm not sure what you were wanting.
Yeah, no, it was a stalked sponge.
Roger.
Don't worry about it, front row. We can keep on going.
the shadow, I think, made me think it had multiple branches. You can see it on this stalk, it looks like.
Oh, we got gat.
It looked like there was an enemy growing on top of it, though.
Steve, another question for the chat. Do you know the depth range of tunicates, going back to that topic?
Well, I know they can get pretty deep, so I would say probably find them down to, several thousand meters, at least 3,000 or 4,000 meters. If you had the ability to look with sufficient effort, you could probably find them down that deep.
Zoom in a bit there if you want to.
Sure. Some details. Glass sponge. There we go.
Another Euplectiidae?
Okay.
Euplectiidae, yeah, most likely. Or, no, that one actually might be a Roselledae.
Oh.
Yeah, Roselledae, Caulophacus.
Ah, it's a brittle start.
That's Euplectiidae there, though.
Oh.
Or actually, no. Hold on.
Polyapica?
Hold on. This one actually might be a Phernomitiae.
Push in a bit if you want, yeah.
Relative of the one we had seen earlier. This one actually might be one that we collected yesterday called Semperella. It could also be Polyapigon, I'm not quite sure, but I think that we have enough imagery here that we can confirm whether it's similar to the one we collected yesterday. I believe it is. So,
And that was a Faraday sponge?
okay. Phernomitiae. Yeah. Phernomatidae. Persingid sea star. Maybe? Can we partial zoom on that one?
Sure, go ahead.
Oh, it's Crinoid. Yep, Crinoid. Very difficult to collect critters. Oftentimes they'll jettison their arms to avoid being collected, and they end up with just a pile of arms on the bottom of the box.
Oh no.
Can we zoom on this other coral while we're here?
Yeah, go ahead.
Yeah, that might be different from the one we've seen recently.
Atalanta heading coming to 180.
So this is more bamboo coral, it seems, or
Yeah, it does look like a bamboo.
it looks darker. Am I wrong in that?
It does look darker, yeah. The pigment could be slightly more dense. I do believe it's probably very similar or the same species as that one we saw just a few minutes ago.
So what causes that kind of denser color and that not as vibrant pink that we usually see with this bamboo
Okay, go on.
coral?
Yeah, I don't have a really good conclusive answer, but there's a few things that could be something related to their diet or you know, their age. I mean, pigment, it could be something that they produce in their tissues that could be used for signaling, you know, to others in their environment. I don't think it's a very well understood, especially for bamboo corals. Pigments are
I'm just on
very,
the high part of the ridge
very,
right now, so.
you know, if you think about the energy that goes into producing different kinds of cells and chemicals, you know, pigments, especially in the deep sea environment where they might not necessarily serve a huge purpose, the things that, you know, look in the visible spectrum, light spectrum. It's a very energetically taxing compound to produce. So you would imagine that there has to be some purpose to it.
Could
So
we
it's a lot more
see
easy
if
to be
any of these
translucent
are
and
okay for
gelatinous.
grabbing around here?
Yeah, I can do.
Here, we can zoom in over here actually.
See something in there you like, do you?
No, it doesn't look very good actually.
Atalanta's sitting at 270 right now.
Yeah, right.
Another question for the chat. Could low oxygen affect these corals as well? What is the saturation here?
Thanks.
The answer is yes. About these corals specifically, I I don't believe oxygen is a limiting factor on their distribution here. Really, we don't really we don't really get talking about, you know, metabolic stress for corals in persistent low oxygen until we're getting down below maybe 50 or 25 micromolar oxygen concentration levels, and right now we're at 117, 116. But different corals will have different, you know, tolerances for oxygen. You know, obviously something that produces a very large skeleton perhaps might, you know, require more oxygen to, you know, precipitate that skeleton. It's a very, you know, metabolically, energetically demanding process that, you know, something that's more gelatinous might not necessarily have to contend with. So, you know, we find that stony corals, I'm going to start mixing up my units here, but stony corals are often not found below like maybe 5 to 7 milligrams per liter oxygen, or rarely if they are, and maybe only solitary morphologies.
Looks like quite a few loose rocks in here if you want to grab one from here.
Yeah. Let's see what looks good. Oh.
Maybe they're loose.
Maybe one of these two right here.
Come down a bit.
Coming down.
Or this guy.
Could happen.
Here's a question for our data logger, actually.
Yeah, this guy looks actually pretty good right
does
here.
Does the data logger also annotate the live dive on the spot, or do annotations come after when the expedition is over?
Currently we are annotating the dive as it's happening, and I believe we do have other data loggers who annotate the videos after the expedition. Am I right, Steve?
Yeah, it's there are entities that do that. It's not a part of the OET or necessarily, but at UH, there's a team that does annotation. Yeah, might get it.
Well done.
Hey.
Nice.
Hey.
It's good looking basalt.
So we have starboard.
Can you look at the bottom of it?
A.
Looks like there's some alteration.
Oh yeah.
Oh yeah, we don't want that.
Oh.
Why don't we want that?
It's highly altered. It's a pelagonite. It's not a basalt.
Such high hopes.
Looks like maybe to the right there would have been some good candidates. Maybe.
Yeah, I mean, I'd be down to check those too.
Yeah, chill.
Although I feel like they're so close to each other, probably similar condition.
We are starting, yeah, we are kind of in this moderate to low slope environment. In a few hundred more meters, we're not going to be going up all that much, but in a few hundred more meters, we'll start to probably come to a rise where we might have other options too. See what we find.
Roger.
Atalanta looking
This
200.
stuff looks all pretty crusty to me. Although there's some
little cucumber
candidates
guy
here.
back there.
Yeah.
Sorry,
Behind the cucumber, there's some. Oh yeah, that one right here, actually.
Are you seeing another rock you like?
Yeah, a little bit below frame.
Roger.
Down here somewhere.
No,
Yeah, this guy maybe.
the square one on the right? That's what you're, or the triangle one?
Let's do the triangular one.
All right.
Oh.
Poke it. Poke it.
Well, if the square one's easier to pick up, we can do that too. Yeah, stuck on there. Probably both stuck on there.
Yep.
Okay. Maybe this pile over here.
My ROV's going crazy there. I'm getting tugged a little bit.
Yep. Those look pretty small though.
They do. Atalanta's gently saying, come along. Huh?
Multiple sea cucumbers here.
Ooh.
Sea cucumber party.
It's the slumber cumber party.
There we go. I knew there was another joke in there somewhere.
Here's a question from the live chat. Why are there no fish around? I haven't seen any for almost all the live stream.
Yeah, the fish are going to be very rare at this depth. We do see them now and again. You know, there are fish that live deeper, but most of the fish that we're probably going to see at this depth are halosaurs or maybe Synaphobranchid eels.
Hit 5 on that bubble count.
But even then, you know, they're going to be rare, sparse, and just not in high densities, much like the corals and sponges.
Okay.
You could infer that if you see lots of corals and sponges, it's probably food is of sufficient quality, of sufficient quantity, that it would support fishes as well, but that's not the case here.
You ready for a move?
Although as we get shallower, I suspect it will change.
Bridge nav. Can we move 50 meters, bearing 110, please?
Pen right here. Can we
Thank
take
you.
a look at that?
Yeah, I can zoom in there, dude.
And yeah.
Is that a hard coral?
This is a sea pen. It's a type of coral. It is one that I have collected on other expeditions, not on Nautilus, but it's good to confirm. But it comes with a brittle star associate. Can we pop a Niskin here? I think it's a good idea. This is the only place we've really seen sea pens. It might be a good comparison since we don't have any sea pen eDNA samples.
Him, or
what's that?
What, just a Niskin?
Yeah, Niskin in this area. You know, maybe no more than like 3 meters off bottom.
Roger that. We are 2.2 at the moment.
And we have all Niskin bottles available.
Yeah, you can hold her up for a minute.
So in this area, we should make a note on the sample that there was a sea pen in the area.
Go for Niskin number 1.
Roger that.
Gibson, here's actually a question for you. What is the yellowish screen on your right-hand side?
On my right-hand side
Must
here,
have tripped it.
Yes.
I think it's a
6,
sub-bottom
5,
profiler
4,
from
3,
Hercules.
2.
So another sonar with different frequencies to profile through the sediment and reflect off some
I can't tell if
more
it
dense
tripped or
material, I
not.
believe, but that's my best stab right now. I don't know if
What's
Steve, you
that?
can help me out.
I can't
1,
tell
2,
either.
3, 4. No, that one's not tripping.
Hangs off the front of Herc and gives us an understanding of the substrate.
You want to go for the bottle 2 just in case?
Yeah. That must be clever rigging there.
That one.
There we go.
Perfect.
So bottle 1 is a no-go.
Appears to be a... Sure. Can go back to 1 on that thing if you want.
Try and find a better ID on that CPAN in the meanwhile.
Something to follow besides mud or sea snow. Sorry.
Possibly Pennatula species? No, that doesn't seem right. No, it's a little bit different than Pennatula.
You're going to go counterclockwise, right?
I am going to go counterclockwise, correct.
Awesome. Wake me up when we get there.
All right. Check out the double shadow you're getting here. Seeing triple.
And here's another glass sponge.
Yep. Yeah, possibly Semperella. This is the kind of environment where I would expect to see nodules similar to what we saw at the other site, but I don't think we've seen any appreciable nodule accumulations at the site at all. But in some ways, I feel like it would be interesting to see when we cut into them today if those nodules are really nodules at all. Some of them appear to be just shards of crust.
Yeah.
Irregular crust. So I don't know if there's another class of nodules, or that's just like uniform or irregular, uniform crust, or irregular nodules. I don't know, somewhere in the middle.
So we have a question again for the ROV pilots. What is the yellow knife-like thing on Hercules? Is that a new upgrade, or has that been there before?
Yellow knife-like thing. Oh, the yellow knife in the front?
Mm-hmm.
Yeah, that is a Green River dive knife made in New Zealand.
Hmm.
And that is definitely an upgrade.
What would be a common use of that
There's
dive knife?
some organism on the left of the frame. Back up a little. Yeah, right here.
Oh, good eye.
Oh,
Oh yeah. Do a quick zoom there.
Sure.
That's what happens when ROVs
Crinoid?
back up too quickly.
So what that is, I think this is our sea pen here, Phoeniculina. But what we have here, this is a deep sea long-spined sea urchin, also known as Espidodiadema.
Espidodiadema.
Espidodiadema, or family is Espidodiadematidae. I think there's not many different genera, but this is one we see quite often. Usually it's a deposit feeder, so it's probably eating, some kind of organic material and sediments. Imagine the long spines probably give it sometimes you find them on hard substrates too, but I would imagine those long spines are favorable for moving across these soft sediment areas.
So what is this sediment actually made out of? Is it made out of shells, detritus? What is it made out of?
So, it's the same kind of foraminiferous shells, but there's organic material here. You can see little bits. Actually, you can see some of the feces coming out of the top. That's actually the
interesting.
The on top is the anal bulb, that's called. So all the food will come out the top after it's been digested. The mouth is on the bottom, and so there's like a feeding trail here of just like fecal material. But yeah, it's probably eating sediments and probably extracting some sort of, nutrition, whether it's microbial or whether it's maybe some sort of, phytodetritus or detritus material that's accumulated in the sediments from the marine snow. Those are all options.
So have you actually taken any samples of this sediment, or we just leave it there?
The sediment?
Yeah. Somebody wants to know.
No, we don't really sample the sediment unless we really need to. If we need to, we can take a push core. But yeah, that's the only way to really get at the sediment in any sort of analytical way.
Okay.
All right, thanks. Good to go.
It's on the move.
It would be hard to get one here.
I'll have to ask Liz if this is something of interest, or if larger urchins are needed. There's not much mass there.
Gotcha.
So Liz, if you're watching, please give us your input.
We see them quite often, though. Even though we've only seen one across maybe 2 days, we see them quite a bit on the seamount landscape. Some rocks candidates here, maybe?
Yeah, I see several in the middle of the screen.
It's pretty sheet flowish to me, but I'll pick one up if you want.
Yeah, not great.
Is that angular enough, or the?
What's that?
What do you think, this one?
Maybe that one, or the one to the left of it?
Yeah, let's do the one to the left of it if we can check it out.
Your call.
Can zoom in there a bit if you want.
Yeah. Can we look at the other side?
Sure.
Yeah, let's take it.
Right. You want to
And
open the
I
box
think
up?
that may fit in starboard bio box A.
And you could hit number 5 there for me if you get a chance.
It would be a good idea to, on that sample diagram, to kind of approximate like the size of them in here, because then it allows you to easily tell if you like, if these are two, like this was a really small rock.
Yeah.
You could potentially double up on this compartment.
Okay.
And save extra space.
Oh, sweet.
So if it's like, I don't know, broad number like 10 centimeters, 20 centimeters, or you know, small rock or nodules or something,
A
just
or
so
B?
they can in A.
Right.
So the other watches will know kind of what's there without having to look through the pages.
Okay. So
This one was maybe about 10 centimeters, 10 to 15 centimeters.
And the other one was also about the same size?
Yeah, it was about 10. Yeah.
Well done.
Not enormous, so easily, you know, you can put two rocks in there, those inboard compartments.
Awesome. Thanks.
All right, stowed. So that is 2,800
Caught
meter
that
rock.
in the Atalanta cam too. It's pretty cool.
Yeah, that was a nice shot.
Thank you. All right, tray closed.
Thank you.
Atalanta heading 115.
All right.
We have another question for the chat. Are those nodules or solids? Or are those nodule or solid on the ridge?
Nodule or solid?
Yeah.
Definitely solid. I wouldn't classify them as nodules.
I was trying for number 1.
So what classifies as a nodule?
Sort of these semicircular to circular class, botryoidal looking, which basically they look like they have tiny grapes all over them. And they come in varying sizes, but
Sorry, couldn't resist.
they're not angular, and they're not connected usually to the rest of the flow.
So which is more common to see life on, the solid or a nodule?
Solid for sure. I don't know if nodules incorporate life at all, besides I guess how they're formed.
Dan, can I throw the Atalanta Zeus into pip H1 here?
Sure.
Those
Sorry,
shadows
looking
get
up
me
there.
every time. I keep thinking that's a black coral.
Honestly, same.
So with some of these coral and some of these sponges, are some of them bioluminescent, or we just can't see them because of Hercules lights, or are they just naturally sort of dark?
They could be bioluminescent, but you would not be able to see that with this light.
One of the blank screens
light
there.
conditions.
Clever.
Yeah.
Do you have PC4 as a source?
Yeah.
Do, let me see. In the bottom left here, at multi-view.
No, one of the black holes here.
Yeah.
That will be
Let's see.
the multi-viewer. I should know this.
Let me see if I can get it.
I think multi-viewer 3, I want to say.
Atlanta heading 140.
Copy. It's the multi-viewer with the, currently has what's probably labeled as Argus cameras in it, or sled. It's probably it's a destination that's, it might be a destination that's labeled as one of the Argus cameras, like Argus Dash. Try Argus Dash or Argus Down, one of those.
Come up a little bit, just watching the sonar.
Right.
That's another one of those tunicates.
Okay, thank you.
Right there. We don't have to zoom in and out, I'm just pointing it out for everyone.
Oh.
One of those tunicates we just collected.
Does seem smaller, right?
It was a little bit smaller. It might just appear that way.
Yeah.
The colony itself, I think, was a little bit larger, but the might appear that way.
To remind us again, for some of the people at home, what classifies as a colony versus a single stock?
That's okay.
Colonies are usually repeating units of individuals or units of individuals.
As a destination?
They typically have,
Dash?
each individual unit, each individual tunicate, may be connected to another, and they might have resource sharing, things like that. But an individual free living, for example,
that as a,
would probably not be connected to another like individual. So there's some sort of
select. It'll automatically go there. Automatically.
There's some degree of cooperation
select,
among all the individual
Dash,
units
for
in a colony.
example, and then source is PC4.
So they're in a sort of a symbiotic relationship with each other? Is that what you mean?
I don't think that can apply to colonies. They're effectively, all the same, working in the same way. Yeah.
I think actually you can also do that.
Usually symbioses have to refer to two, different either species or groups of animals or something like that.
Or was that another crinoid?
Since they
look like
do different things, but they have a shared
shrimp as well.
Shared interest, whether it's one parasitizing another or one helping one another for some sort of mutual gain.
So on these dives,
sea cucumber.
Yeah,
beautiful.
Large sea cucumber. I think it's similar to one of the ones we collected yesterday as well.
Yeah.
The one that came up, I think, in the best condition of them all.
Well, didn't we almost get one yesterday, but it floated out of the...
Yeah, that was sad.
Oh, man.
The current snatched it as they were about to drop it in the box.
Oh, no.
So speaking of this life down there, it doesn't seem to be a whole lot. Somebody in the chat wants to know, are you surprised with the lack of life that you see in these dives?
Destinations are always red.
This is what you were anticipating?
Yes and no. So yeah, I'm kind of expecting this type of setup. It's very similar to what we saw yesterday. But I know that as we move shallower, as we saw yesterday, the diversity will start to explode. Yesterday that happened around 2,400 meters or so. And we're going up to 1,000 meters shallower than 2,400 meters, so we should see a lot more species.
Like
but the interesting thing is that,
like that.
even below 3,000 meters, which is kind of our goal for starting many of these dives, it's really not been explored below 2,600 meters in this area, and especially below 3,000 meters. So being able to go that deep provides us true exploration of this area and the depths we find around Johnston that no one's ever been to.
Yeah, sometimes I forget that we are the first people actually seeing this kind of, this ridge in this area.
Right, yep.
And so are you, viewers at home.
And yeah, and from a geology perspective too, the deeper you are, the more likely you're going to get, the original volcanic basalts that compose the seamount too. If you dive shallower, you might be getting carbonates or, something that might not be useful for accomplishing our geological goals.
Oh, we have a question for cameraman Dan. Question for the video engineer. Is the zoom and focus are controlled by physical buttons, or is it more of a software kind of deal?
Sorry, could you repeat that one more time?
Question for the video engineer. Is the zoom and focus, are they controlled by physical buttons, or is it more of a software kind of thing?
It could be either or. So it is physical buttons. We actually have a couple different panels up here. One that is dedicated solely to zoom, so that gets us from our wide to our telephoto, and then the zoom control is on a slider. It's really sensitive, and it's especially difficult when you're trying to focus on midwater things that are kind of floating in and out of the focal range. And then the iris is really what's controlling your brightness here, and all of it can be on auto, but the reason that we do full manual for everything is usually it does a better job when you're trying to find when you're trying to lock onto specific animals. Sometimes it'll want to expose for the sediment rather than that animal. So
we've seen this bamboo
always running in manual
quite a bit. Can we do a small snip of this?
You want to do a snip of it?
Yeah.
Yeah, copy.
Maybe just where it bends. Snip and slurp.
So
I love the combo.
If you have time.
Yeah,
we'll see another. We've seen at least half a dozen in the past hour or so.
For a second, Dan. And Gibson, if you could show me the hit 5 on that thing.
When we see these, bamboo corals are pretty difficult generally to identify, but when we see these enough,
you want 5
as the kind of dominant coral for this area, it's worthwhile getting a snip to confirm identifications. And I think this will be our first coral collection of the cruise.
Is this going in the suction or in the...
Yeah, it'll be we'll do a snip and slurp so you can get the jar camera up there.
Roger that.
Out there, Steve? Or?
Looks good. Even that's plenty. Yeah, more than enough. Yeah, that's good.
Good chunk.
Okay, well, let's do this.
75%?
Sure.
We have another question for Steve, whenever you're ready.
Sure.
Do these sponges have any natural predators? And do the sponges have any natural defense against predators?
Sea stars or sea urchins sometimes. And I'm guessing some of them probably do. Or some of them might be less palatable than others. But in terms of like active defenses, I'm not quite sure. Sponges don't have a lot of nutritional value, but when you're down here and it's either sponge or mud.
There we go.
Perfect.
Which would you rather eat?
In the jar?
Oh,
Okay, you
that's
can
fair.
kill it. Move
The
a
sea
jar.
urchin wasn't eating, it was eating the sediment, so
Yeah.
I guess
Got it.
first the mud.
Lovely.
Okay, you can go wide
For
there,
personal
Dan.
connections, because I grew up with SpongeBob, I'll take the mud.
Good job. Great collection. So that was a bamboo coral. Chyrsogorgia.
There's your 6.
5, 6. Come on, 7. Lucky 7.
Another question for the follow-up. What creatures will you and won't you take for samples of? Is it just invertebrates? Would you slurp a
Gonna
fish?
hit one on the...
We can try to slurp up fish, but oftentimes it doesn't work. Yeah, they're too fast. You know, once in a while we'll get fish. For example, if we're slurping up, you know, I remember we got several fishes slurped up many years ago when we were working on hydrothermal vents where you have large shrimp aggregations, vent shrimp aggregations. You'll have snailfish and things like that in there. Sometimes you'll slurp one of those up inadvertently, but fishes in the water column rarely get sampled. In one instance, I can remember we had a long eel-like fish called a halosaur get stuck in the fibers and cables in the ROV.
Oh no.
And it kind of got stuck, and that was the only way that was able to come up. But that was not this ship and not this vehicle.
So when you get these fishes that come up, or if you do, what is the protocol for it? Do you preserve it? Do you put it in a tank and then ship it back to the mainland? How does it work?
So the fishes that come up will probably not be very intact, since they have gas-filled sinuses in their body. That those air bladders will expand and cause barotrauma, unfortunately, which is why we usually don't get fish anyway. They don't survive. The red sea pen just to the lower right, just off the right corner too. You can do a snap zoom on this one, since it's a different species. This one is probably in the Protoptylidae.
Sort of dusty. Sorry, Steve.
Oh, that's all right. You got it. Okay, look at it. At least for video. I don't know if I got screen grabs.
Can now wait for a minute here. Come on.
Yeah, so if fish do come up, we'll preserve them kind of in our typical fashion with either ethanol or formalin as needed. But we try not to sample fish if we don't have to. Most of the time we can get really good identifications with imagery if we're careful.
There again, Dan.
This is a different species of sea pen. I don't think we saw this yesterday, but this could be in the family Protoptylidae, or it could be a different family. Could be
Okay, check's complete. Looks good.
sea pens. Cophobelemnidae too.
Go a little tighter.
Yeah, maxed out there.
Oh, okay. Too far away.
What do we got for time?
30-ish minutes.
Yeah, are we stopped? Can we sample this?
Yeah.
Pull back.
Ooh.
It looks a little bit different than the Protoptylum that I'm used to seeing around here, so it would be an interesting collection. This one is probably going to be might just be a slurp.
Yep.
You could do like a grab and slurp too. I'm not sure if it's going to run away on us.
I don't know if I could get that
It's
low.
going to pull a sea cucumber.
You want the slurp on right now?
No.
Okay.
It's a very small one. So that it's between two families suggests that this would be a good collection to clarify. It also helps us, you know, we just took an eDNA sample just a little while back over soft sediment. It would be interesting to see if we can detect this species in our eDNA sample as well. So this would help ground truth that, that methodology.
Okay, Dan, you want to zoom in there?
So, someone in the chat wants to know, do we bring extra amounts of preservatives and alcohol for the sea sponge or the sea cucumbers that get stuck, or the other specimens that get stuck to the ROV?
Well, I mean, we treat everything the same effectively.
Look at that tornado going on.
If it happens to come up on the vehicle, or if it happens to come up on in the boxes. So we have plenty of chemicals to deal with both of those scenarios.
75%, Roger.
Another person also wants to know, from Bill from Sacramento.
Almost got it.
On this expedition, will we be seeing any ships or any shipwrecks, do you think?
Likely not.
Okay.
Oh, well.
When do you want me to?
I think it might be. It's one of these that you can chase them all day long, but they just keep on going down and down. Although, you know, the way that the sediment's moving, it gives you an idea of how.
I think we're getting to the end of the tether here. Want me to turn auto heading
off? It's not very
off?
viscous at all. It's pretty loose, fine stuff.
Auto heading off.
I think it would end up wasting a lot of time if we don't have it in the bucket now. You know, don't worry about it.
We'll find out sooner or later.
All right.
The tug, anyways.
0%.
No, you can keep running.
The way it escaped like that suggests it's probably Protoptilum or something in the Protoptylidae family, in which case I do have bits of this family, possibly similar species.
Oh, I think we got something in there.
From the regen. Okay. But it's one of the few sea pens that just disappears. They have such a very fast reflex of being able to get down to the sediment.
Does these type of coral that they can move? They can. Is that what you're saying?
Yeah, they have the bottom of them of these sea pens has like a peduncle, which is basically like a balloon that they can inflate and deflate, and much like a clam would, you know, inflate and deflate, or you know, a clam would use its foot to dig into the sediment, right? These sea pens can do something similar with their peduncle.
Ah. Is this with all types of coral or just the sea pen?
You want to turn the camera?
Not all sea pens can do it, but all sea pens have peduncles or something like a peduncle. So they can't all escape.
Oh, no, you leave the camera on the door. Sorry, we're not done yet.
We have another question for the chat for ROV pilots whenever they're ready. What type of maintenance does Herc need during an expedition?
Lots and lots of love. Constant maintenance.
Encouraging words.
Are you going back for more?
No, I'm trying to catch up to you.
Okay.
But we want to... Oh, we got him. We got him.
Hey!
Oh, we got it?
Oh, I got a piece of them in there anyways. See it?
Yeah.
Nice.
Yeah, we'll log it as a sample, see what it looks like.
Show me the hit number 5 on
there might be some other things in there that would be interesting, maybe some mollusks or snails or something.
And that is in jar 2, am I correct?
That says...
Yep, jar 2.
Yep.
Awesome. Thank you.
Need some new glasses.
I need some new contacts.
Sure.
Staring at these screens all day. My mom worried me about this.
Okay, you can rotate the jar now to jar number 1.
Rotating jar.
I think, yeah, if you're not already, you can also come
so
up.
we're going to go with Crinoid for that family. I can spell it out in the chat. Do you have the chat window open over there?
Yeah.
Okay. I'll just put it in the chat.
I'll just come under you here.
Tentatively.
So you can keep your heading.
Okay, roger that.
Oh. Thank you. Awesome.
We have another question from the chat saying,
there we go.
How do you preserve the sea cucumbers? Since they're mainly made of water, I would imagine it would take a lot to dry them out or preserve them. How do you do that?
Come back down a bit.
Yeah, so we use a couple of different methods depending on what the goals of the science we're trying to conduct calls for. Oh, there's a different glass sponge. Most of the time with sea cucumbers, yeah, it depends on what you're trying to do with them. So if you're just trying to get them into a museum, repository with the most available, means for scientists to look at their and identify them, you probably want to use a couple of different preservatives. You will want to take a small portion of the sea cucumber and put it in a very high concentration ethanol, either 180 to 200 proof or 90 to 95 or 100% even, absolute. That preserves the DNA in the tissues. So you can use things like genetic barcoding to identify it or characterize it molecularly, phylogenetically, or you can also preserve the sea cucumber in a concentration of formalin, which is basically formaldehyde, which would firm up some of the gelatinous parts of the tissue. Not much still, but it would help a little bit. So formaldehyde would harden the tissues and allow you to properly transport it more easily without breaking itself apart.
Atlanta heading 015.
But that wouldn't necessarily preserve the DNA very well. So we use a combination of methods. There's a third option. Oh, there's our fish. There's a third option, which is just freezing it, which helps to preserve it relatively intact. So it can be thawed and examined later, but we rarely will go with the freezing route because of the challenges with shipping things cold, from Hawaii to museum repositories often.
So the best sort of thing to do is just put them in formaldehyde or alcohol and let them sit.
Yeah. The formalin treatment will usually last sometimes 24 hours to a couple days or a few days. And then they'll be transferred after that to a lower concentration of ethanol, maybe 70% or so for storage, which helps inhibit any bacterial growth.
And I think that fish we just saw might have been a Basalzetus.
Yeah, it could have been. Yeah, that's a distinct possibility. Oftentimes with fishes, it's very difficult to get good imagery. It takes some fancy flying because they really like to orient away from you. And so you have to kind of rotate around them, but also make it look like you're not moving around them so they don't get start to move on you.
I see.
I was up against the hill there and I goosed it a little too hard and blew it out.
So if our listeners still listening, and they asked earlier about the required maintenance, I'm a little less preoccupied. So Hercules is part electronic, part hydraulic, which means fluid power, and awesome mechanical components too. So it needs maintenance on all of those things. And when you're going subsea, it needs additional work, sinks and anodes to prevent corrosion, and cleaning up, flushing fresh water. I'm slowly learning those steps day to day here. And then as stuff needs replacing, we get into the ROV shop on the Nautilus and get to work.
So, about how long do you spend in the workshop working on Hercules, working on Argus, working on Atlanta? Like, about how many hours a day would you say?
4 hours minimum between dives. That's a busy 4 hours.
So then, would you say that you have any off time at all in between these dives, in between working on Argus and working on Atlanta and Hercules?
No, not a lot. Enough time to eat and get a minimal amount of sleep.
Do you like working on these kind of things? Is it like a rewarding kind of challenge, or is it just going through the motions?
Oh yeah, it's a blast.
It's a total blast. If you're into turning wrenches or geeking on electronics, it's paradise for us. Been doing it for a while and it never gets old. There's always a outstanding maintenance list that runs. There's dozens of items on there. There's long-term maintenance, short-term maintenance. And, there's always projects to pick up and run with on the ROV system. Not only the ROVs, but all the ancillary equipment that we have, the cranes, the winch, all of our spares, consumables, tooling and such that goes on the ROV.
So how long would you say have you been working on specifically Hercules and Atlanta?
I've been coming out with this system since 2018.
And do you know when we actually acquired these types of ROVs?
The ROV was this ROV was built in 2000, right around the turn of the century, 2002. I think they finished it. And I started doing sea trials with it. So it's
turn of the century.
20-year-old vehicle.
Yeah.
You think about an automobile that was, 2000. Like I have a truck that's 2006. It needs a little more maintenance than it did 10 years ago.
Yeah.
If anyone watching at home has a car from 2000, write in and tell us how it's doing.
I mean, I have a car from 2008, and it's kind of crappy at this point, but...
It's as old as you.
My first car was a 98.
That was the year I was born.
It gets to a certain point when your car's older than you are, and then you get it's a little odd that you're driving something that's technically older than you.
My first car was a 96 and it treated me beautifully. Maintenance was the easiest.
My first car was a 06 and it left me stranded in Kona. But she still works now.
So stay pre-2000s is the lesson
There you
there.
go, that's the lesson.
I'm going to date myself here. My first car was a 67 Volkswagen and I still have it.
That's cool. Wow.
It's vintage, it's vintage.
Yeah, what we're doing with Hercules here would be akin to getting into that turn of the century automobile and driving at a high rate of speed across the country. And the only spares and tools
are in the car with you.
Are in the car with you, or in the trailer you're towing behind it.
So is it just, this is just constantly upgrading a Hercules, or is it most of the same systems from 2000 and some odd?
It's both. There are components, for example, the electronics that the brain of Hercules is still the original 386 based PC-104, which is now obsolete. So they're no longer manufactured. We have spares for those squirreled away. So quite a bit of the spares for the original build we still have, and we carry those on board, carefully packaged and stowed in a temperature-controlled environment. But there's constantly new components on the vehicle. For example, there's a new sonar, forward-looking sonar. There's a couple new cameras on there. Definitely a lot of new lights. We're constantly going through one of our biggest maintenance issues on any ROV is the cabling. So we have a lot of rubber molded cables with gold-plated connections, and we try and keep the seawater off of them for obvious reasons. But the cables, they're in, we constantly changing them out as they get old and start to give us issues. You'll hear us talk about ground faults once in a while, so that's, the electronics are coming in contact with the seawater. Bad things happen.
So out of all of our ROVs, which one is like the newer model, or the more up-to-date version?
Atlanta is definitely the...
And when was Atlanta made?
I'm not sure when that was made.
And then going off of that, somebody in the chat wants to know, how old is the actual EV Nautilus?
The hull was laid in 60, I want to say, same year as my Volkswagen, 67.
Oh.
Oh, is that another spinning snail?
Looks like it.
Oh, nice
Look at
eyes.
it move.
This is where it's tumbling down the ridge, like you said, Steve.
Mm-hmm. Yeah, that's what they do. You never know where they come from either. They're always
How'd
moving.
they get up there?
You never see them just sitting on the seafloor.
That's a nice surprise.
It's like a tumbleweed of the ocean.
Tumbleweeds, yeah.
Do they just keep going down?
Until they get stuck. Like now it just got stuck here.
Oh.
Can zoom in a bit there.
Yeah, yeah. Let's see here.
Hiding behind the rock.
Yep.
Yeah, it's stuck.
There it is. Yeah.
Yep.
Or maybe it's just taking a nap.
I like that option.
I have to rest after all that exercise.
Tumbling's hard work.
Okay.
Yeah.
We have another question for the ROV pilots. Do we track the mileage or hours of Herc?
Absolutely.
So how many miles would you say Herc has gone?
Well, we track the number of dives that it's done, and then each of those dives has a number of hours associated with it. There's an hour meter on our power distribution unit, so we know how many hours the motor has been spinning. So the electric motor and the associated hydraulic pump are both dynamic, they're like your car motor.
Sweet. So then, like, at what point, because because eventually with this much, I assume with this much strain and effort that Hercules goes through on a day-by-day basis, at what point do you have to do like a complete replacement of parts?
That's an interesting question. At the moment, we are in the process of designing a replacement frame and a flotation, which is a major component of the vehicle. It'd be like taking every single component off your car and putting a new frame on it and then putting it back together. So that's going to happen for 2023.
Okay.
The flotation or the buoyancy blocks, the big yellow blocks that keep it floating here, are the original. So they're made out of syntactic foam, which is micro glass spheres and epoxy. So after years, that starts to delaminate a little bit, and there's a few cracks in it, so it takes on seawater. So we don't have quite the buoyancy that we used to. So we'll replace those as well.
So is it just a constant like upgrade and a constant like replacement of parts, or will we actually replace Hercules entirely with an entirely different ROV?
I think that's in the cards, but building a new vehicle is it's way above my pay grade.
And then we have another question of Herc. If you could upgrade anything at all about Herc with no limitations, what would you upgrade?
Everything.
Everything. Anything specifically, like controls or camera quality or cup holders?
Yeah, yes to all those.
If you gave it a bigger foam pack, it would get heavier and you need a stronger crane, but you have a bigger payload and could maybe fit some more tools in there. It would be interesting.
Specifically, I would, so we have the one HD camera on here, the beautiful one that that you all see the view that goes out. But our other cameras are still standard definition cameras. So I would upgrade all those.
I'm just coming up, keeping my eye on the ridge behind me.
Yeah, Reg.
Yeah, imagining this in 4K would be stellar.
We have a 4K camera for the system, but there's some complications with just slapping that on here. You have to get that 4K signal up to the surface, which we can do. We have a spare fiber. But then once it comes to the surface, then we need to record the imagery and then process all of that. So that's...
well, not only the data, but do you have issues with overheating the titanium housing and all that?
Yeah, yes and no.
Yeah.
The 4K camera that we have is actually, by today's standards, it's stated as well. That technology is moving pretty fast. I would settle for just HD. It's,
right.
For example, our...
on the side of the ridge I want to be on right now.
Our bubble camera is, that's like, 90s technology. It's 45-degree viewing angle, which is nowadays they're like 70 to 90 degrees.
Yeah. It's the one on screen now for all of our viewers. Just changed on Sat 3.
We have another question for the chat saying, how does it feel to have one of the coolest jobs in the world?
It's a great feeling. Never gets old.
Yeah, thanks, Lynette. No, it's a pretty awkward position right here. I'll start pulling you up the slope, Dan.
Yeah, Raj, there you go. Less talking and more flying.
Another question. whenever you guys are ready. Will the frame and the foam upgrade change the depth range of Herc?
Oh, what is
Oh,
that
something under the rock.
gelatinous
Is that a
thing?
flatworm?
I think it was a tunicate,
That was a tunicate.
one of the predatory tunicates.
Sorry, I'm catching up here, Steve. There is talk of increasing the depth with the foam. There's trade-off there. It'll be heavier. But it's possible we might be able to increase the depth capability a little bit. It's currently, our top scientists are all over that.
So with Herc, are there, as it does sustain an immense amount of pressure, are there any specific parts that are meant to bend, meant to compress to help compensate for it?
Mechanically, not really, no, but there is some, or the compensators for all the hydraulic fluid and the compensation fluid. So those are basically, if you could, what's a good analogy? If you take a balloon and you blow it up, put air in it, and then you take that balloon and jump in your pool with it, the pressure from the water as you sink down in your pool will put pressure on the inside of the balloon. So we use a similar technique with our with the fluid, all the fluids in the vehicle, but they have a spring that's pushing on the bladder, which is open to seawater. So that keeps our hydraulic oil and all of our static oil that we use to compensate hoses and J-boxes just above ambient seawater pressure.
Oh, sea cucumber. So you'd say it's more of like a rigid structure that's just supposed to stay strong rather than flex and bend?
Was Liz done collecting cucumbers?
No, we can, how do you feel about pausing here for the watch change and doing a quick slurp of this cucumber?
Yeah.
I think she would love that.
Okay.
You have 8 minutes till top of the hour.
Yay, we can eat.
Oh wait, there's actually there's one of those zoanthids here too.
Oh, I see him.
Yeah, you see this?
How's the delta for you, Dan? Is that good there?
Rather have him.
Too high?
Can we get a zoom on both of those? They can get the zoom after, I guess, after we do the slurp.
Yeah, I'll grab the, can you show me F5 on that?
Yeah.
And a jar as well.
And a jar, you bet. On that, let me know if you're concerned with the...
What do you do with all these?
The photos?
Yeah.
They go into the cruise record, the hard drive for the cruise.
Do I have to do anything, or is it?
Nope, it's automatic. You'll find that folder will empty every.
Have much room to come out before I pull on
probably every hour or so, and it'll be backed up, and then it'll be
a little bit.
Clean again.
You want to zoom there, Dan?
Yep. So this looks very similar to a sea cucumber called Benthic.
75 coming up.
Too big.
75%.
There you go.
It's going to be fairly rigid.
It's quite rigid,
If this one doesn't go, we'll put it in the forward box.
Roger.
Forward box.
Maybe lambda.
I might try to make a run for it like
You wanna
last
put
time.
that out?
Open the forward box. Yeah.
Forward box going out. Oh, I'm going to hit you there.
It's all right. Good. It's me backing up.
All right, we'll check it out.
All right, it's fully out. Argus depth's getting down to 10 meters.
Yeah, Raj, I'll come up here. Okay, can kill the suction.
Okay. Suction off. Jeez.
What?
Gentle.
Gentle.
And it made it.
Bingo.
Nice.
Okay, close the box.
All right, I'm winching up slowly too.
All right.
Nice.
It's a delicate maneuver. All right, box is closed. Thrusters. Oh. We'll get rid of the bucket. Yep, thank you.
Okay, can you come up?
Coming up. Delta's at 25. I got 22 altitude now, though, so I'm going to let you come up.
Roger.
Okay.
All right. That's a little delicate. Come off SPL first.
No. They zeroed 6-8 reps in the dive? Okay. I'm not too worried about 6-8 reps. Yeah, I'm just trying to catch up here and get on the other side of you. So you're going to turn to port, I guess? Probably just that tail of the ship there, so something like 135-ish. Is that about right? I can't hear you muted, Emma.
Every time.
I'll keep telling you.
Sounds good. So they just moved back, so I'm gonna move us up this ridge soon. But I think that you still might be getting settled in about them moving back.
Okay, is the ship stopped right now? Or
Yeah.
Oh, it has. Okay. Okay. Should be in a little better spot. I'll let you kind of get sorted out. Sorry, gotta get my feng shui going here. Have to have things all in the right places.
Don't worry, I have my globe for navigation right here. Is the back row, do you guys want to start moving up this ridge? Yeah?
Yep, I'm all ready to go. You guys ready?
Yeah.
Awesome.
Yep.
All right, how are we feeling about introductions? You want to start?
Bridge nav.
Yeah.
Can you make a course of 080 at 0.2 knots?
Roger, 080.
Mm-hmm.
So it looks like we're headed up a ridge of pillows, a lot of pillows. I'm not sure if they're in place or not. A little lightly sedimented. Some of these things could be in place, so I guess we just keep on looking for biology and...
little fish.
Oh, there's fish, yeah. And no hurry on a rock sample yet.
Nice. So, aloha kakahiaka. Welcome to Nautilus Live here from our morning shift in Johnston's Atoll. I wanted to, while we're getting all settled in here, if we could kind of start around the room and give short introductions for this morning so we all know who's talking today. Rob, would you like to go ahead and introduce yourself?
Sure, I'm Rob Piccolini. I'm the watch leader and a geophysicist and geologist. Don't always like to touch the rocks, but I'm enjoying it this time. I'm work at the University of Rhode Island, but I'm from the thriving cultural metropolis of Portville, New York.
And you make delicious food. Thank you, Rob. Liz, how about you?
Yeah, hi, I'm Liz Miller. I'm a guest scientist here. I'm from the University of Hawaii, although I'm originally from Seattle. Yeah, excited to be starting this morning.
Mahalo. Thank you, Liz. Taylor, you're up.
All right, my name is Taylor Ann Smith, and I am the science manager aboard the Nautilus and currently acting as a data logger here on this watch. This is my 4th season on the Nautilus, and I am a graduate student at Cal State University Northridge,
Awesome.
doing some research on microplastic pollution in the deep sea.
Thank you so much for doing that. Mahalo. Okay, front row, who's up next? Emma, navigator?
Hi, I'm Emma. I'm from Annapolis, Maryland, but I go to the U.S. Coast Guard Academy, and I'm a navigation intern on board the ship.
Thank you, Emma. Josh, you're up, pilot.
Yeah, I'm Josh Chernov, and I'd like to ask Dave for a nice zoom on the sponge, please.
Yes.
And I'm your Herc pilot for this watch. I'm the ROV operations manager for OET, and I'm hail from Vancouver Island, British Columbia.
Thank you, Josh. Kylie, how about you?
Hi, good morning. I'm Kylie Pasternak. I'm an Argus pilot, and I am here. I'm here.
You're awesome, Kylie. Okay, Dave, how about you?
Evidently, that's all Kylie's going to vouch for this morning. It's just presence. Not volunteering anything else. I'm Dave Robertson. I'm video lead for Nautilus this expedition. And I hail, like Liz, from Seattle, Washington. But I spent most of my time living in Anchorage, Alaska. My wife and I split time between Anchorage, Alaska and the coast of Oregon, where we have a house as well. So I live in Alaska, I live in Oregon, I work in Hawaii, and when I'm not on the ship, I'm retired.
Hey, wonderful. Awesome, you guys, thank you so much, and I will close out our introductions. I am Hokupihana from the Big Island of Hawaii, and I am here as the Nava'amao Marine Stewardship Executive Director, so we use canoes to care for our oceans, and I am your science communicator today on watch. Mahalo for joining us. Malama.
Yeah, there's something inside.
Yeah, a couple of guys.
Maybe
Yeah, there usually is. That's where I was going for, to see who's home.
Good shot.
Yeah, that's great.
Okay, come wide, please.
Thanks, Dave.
Laser's back on.
You could do a reset on that, I think.
Sounds good.
It's a little, I mean, it's a tiny bit off. But.
Oh, that was pretty far off.
All right, you guys, so we do have some questions coming in. If anybody's ready, I'd like to start with just, let's see here. Oh, so Liz, I'm going to shift this one over to you. Have you ever dropped or lost a specimen?
Yep.
Yes.
I have.
If you were watching yesterday, you might have seen it. We're collecting one of those cucumbers, sea cucumbers, and they're barely negatively buoyant. They really like to float, and so sometimes when you open up the basket and you drop it in, they don't go straight down.
Right.
Some of them can actually swim as well, and we lost one yesterday, but it's okay.
Hey, right.
So yes.
So yes, it does happen, and we just power right through it and move on to the next.
Yep.
So this one, I'm going to direct it towards our pilots here. How many hours would you estimate you have been piloting Hercules now?
I don't know. I didn't keep track.
A lot.
Hercules specifically, I don't know, maybe a couple thousand.
Wow.
Trevor's got that
Trevor has a
Excel
great
sheet for everything he's done, but
I know there was another system that I ran a lot over about 5-year period, like every day for 6 to 8 hours a day, 5 days a week.
Wow.
I think I racked up over 5,000 hours on that machine.
Oh man, you have good driving skills. ROV piloting skills. Nice. Thank you, guys. And this is, I'm just going to open the floor for this one here. In regards to that sponge that we just saw, somebody wants to know what kind it is. Does anybody know what kind of sponge that was?
some kind of glass sponge, but
the ID is a saccocalyx sponge, the one that looked like a rose, kind of.
Yeah.
Yeah, saccocalyx is. Let me see. What's the full name? Ooh, Euplectiidae. Saccocalyx. I cannot pronounce that.
Is there a common name for this sponge?
No, 80.
Not listed here on this guide, but
fluffy rose
only 100 degrees.
yeah, it's really pretty though.
Is it? That was really pretty. I was wondering what was inside. Okay.
Yeah, it looked like there might have been some isopods maybe inside. I'm not sure.
Roger.
Yeah.
Cool. Yeah, I couldn't really see super close. Oh, what is that? And then now, Liz, this one is specifically for you. Liz, what animal are you hoping to see at this depth? You can't say sea cucumber.
I think she has to.
That's usually what I'm looking for. But I don't know, there's a lot of other stuff that's exciting to see. I mean, everyone always loves to see an octopus. Something like that would be cool.
I'm going to do a gauge check.
But it's all, I don't know, it's all exciting.
Right. One of the kids today in my talks asked me if I had seen a giant squid. And I was like, no, but I would love to.
Very
we did
few
see a
people
squid.
have.
It wasn't giant.
Yeah,
All right, you guys,
awesome.
so here's a no pressure question. What is one thing you would upgrade about Herc if you could? Your answer is being judged against the last watches. No pressure.
That's funny.
Well, considering that Josh is in charge of that, I believe that he should go first.
Yes, Josh.
I want to hear everybody else's answer first. I want to go last.
There's been a lot of, I know a student asked if there was a microscope on Herc.
I mean, one of the things I was talking to Josh about before was to have an ADCP, a current measurement system on here, just to see what the actual currents are.
That would be cool. I think currents have come up a lot. Yeah.
Yeah.
Any other new additions, upgrades?
I'd like to hear from the scientists mostly, because that's where I get a lot of my feedback. So
Okay,
I am
Liz.
curious to hear about this.
I don't know, I think maybe just additional ways to collect really delicate animals without hurting them. Maybe a larger slurp.
Yeah, a larger orifice or something.
Yeah.
We have played a lot with these little soft grippers.
Yeah.
Kinda cool.
Squishy fingers.
Those are
squishy
cool.
fingers. Yeah.
Those claws, they can really do a number on some animals.
Yeah. Yeah, we played with these ones that were they were soft, and then they had pumped in, when you grabbed the specimen, you pumped water into the fingers and that hardened them. But they had these soft grippers on there, pretty cool.
Yeah, that sounds pretty cool.
More cameras.
More cameras. You can never have enough cameras.
Never have enough cameras.
What about sampling containers? Would you guys like more sampling containers?
I mean, always.
Always, right? And then we have sediment core samplers and water property samplers, Niskins.
Zoom in, please.
Oh, would we has anybody ever gathered plankton can Herc collect plankton?
No, we often would do tows from the ship, I think.
You would do the tows?
Yeah.
There are much more, efficient
More
ways
surface?
of collecting plankton.
There's no deep sea plankton that come down except
There
for
are.
the snow?
There's a lot of plankton.
There's the squat lobster.
Oh, yeah.
Is there?
Yeah.
Okay.
Ooh.
There's always one in there.
Darn it.
Chyrsogorgia.
Where's the lobster?
Squat lobster right in the middle. See it? He's moving his little arm there.
Oh,
Claw.
cute. Okay, so now we have a lot more questions coming in.
Go come wide, please.
You folks let me know when you're ready. Oh,